Hello everyone, my name is Alejandro Sosnik and today we'll tell you about the research that we are conducting in my lab. So nanotechnology has made a significant contribution to develop advanced drug delivery systems. The idea is that we can combine different types of drugs with different types of disadvantages, for example, poor water solubility or the incapacity to cross different types of biological barriers in our body with nanoparticles, and by doing so to increase the delivery of the drug at the target body site. So in my lab, we are combining different types of biomaterials and strategies to develop advanced drug delivery systems. So we are, for example, processing pure drugs. We synthesize and characterize different types of polymers and combine them also with ceramic materials. We produce different types of nanoparticles and microparticles. We combine them in more complex drug delivery systems. We also use different types of technologies to develop methods that can be eventually scaled up under the industrial setting. And finally, we conduct a broad spectrum of biological studies by using different types of, for example, 2D and 3D cell cultures and also animal models. So to give you an example of the kind of research that we are conducting today, I will show you a project that is currently running in my lab, which is at the interface of nanobiomaterials and sonodynamic therapy. The idea of sonodynamic therapy is that we can take a nanobiomaterial, we can inject it into the vein. The nanoparticle will be accumulated in the tumor by size or different types of modifications that we can do on the surface. And once we have these nanoparticles accumulated in the tumor, we can come with, for example, therapeutic ultrasound, irradiate the nanoparticles locally, and the nanoparticles will produce reactive oxygen species which will kill tumor cells. So a few years back, we found a very simple way to produce this type of nanoparticles. In our case, we are using hybrid titania polymer nanoparticles which can be tuned in terms of the size by changing, for example, the production method. And we can not only encapsulate hydrophobic drugs, but we can also, by changing the size, optimize the accumulation of these nanoparticles in different tumors in a more personalized manner. After characterizing the nanoparticles with a broad spectrum of advanced techniques that we have available in our faculty and also on campus, we demonstrated that these nanoparticles, once they are irradiated with therapeutic ultrasound, produce reactive oxygen species which can kill cancer cells. In the next stage of this project, we are going to begin the feasibility studies of this approach with these nanoparticles in the relevant animal models. So if you are interested in this project and other projects running currently in my lab, you are more than welcome to contact me in the near future.